So we can pass to the second speaker, SARA BUEZO Montero. She is at third year of PhD at Sapienza University of Rome, and she's going to present validation of human IgG response to Aedes salivary proteins as a novel marker of human exposure to Aedes albopictus. Please, Sarah, you have 20 minutes.
Okay, thank you. Okay, hello everybody, and thanks Elena for the presentation. I'm going to present the results that I obtained during my PhD thesis. The title of my presentation is Validation of Human IgG Response to Aedes salivary protein as a Novel Markers of Human Exposure to Aedes albopictus. In the last decades, Aedes mosquitoes have been responsible for an increased transmission and several outbreaks of arboviral diseases as dengue, chikungunya, Zika, and yellow fever, creating a renewal challenge for public health. Aedes mosquitoes are impressively expanding the geography distribution, especially the tiger mosquito Aedes albopictus. Tiger mosquito is the main vector for the European outbreak as dengue in France and Croatia in 2010, France 2013, 2014 and 2015, and the autochthonous cases in Italy this year. It is responsible also for the chikungunya outbreak in Emilia-Romagna in 2007 and Lazio in Campania in 2017, as well as Zika in France last year. The main method to limit the transmission of these arboviral diseases is still to control mosquito vector populations and prevent the contact with humans. Currently, classical entomological tools are the main method to evaluate human exposure to Aedes mosquitoes. However, entomological indices have some limitations and they bring an indirect assessment of human vector contact. So due to this limitation, explanation of human immunoresponses to mosquito salivary protein is emerging as a relevant additional tool. Serological tools bring a direct measure of human vector contact and they can assist of effectiveness of vector control interventions. In fact, while feeding on their host, mosquitoes inject a cocktail of salivary proteins, saliva or blood feeders evoke invertebrates, an antibody response that can be exploited to evaluate vector-host contact. However, saliva is a mixture of several salivary proteins, some of which are widely spread among blood-feeding arthropods, and this may give potential problem of reactivity in immunoassays. So thanks to the large amount of information from transcriptome studies on salivary protein repertories of blood-feeding insects, highlighted the existence of several family and genus-specific salivary protein, which may represent ideal candidates as marker of host exposure to specific mosquitoes. A solid proof of principle has been provided for malarial vectors, and my PhD project is focused on finding similar biomarkers in mosquitoes of Aedes genus. One of the culicin-specific family of salivary proteins is the 34K2. We were lucky because our collaborator Paolo Gabrielli and Federico Forneris at the University of Pavia optimized conditions for expression and purification of the Aedes albopictus and Aedes aegypti 34K2 salivary proteins. And we checked the suitability of R34K2 as novel candidate of host exposure to Aedes albopictus. And we also checked the potential cross-reactivity between R34K2 and AE34K2 because they share a 62% of amino acid identity. Experimental plan started with the first validation of R34K2 and AE34K2 antigens in a murine model. It allows a controlled exposure regimen to mosquito bites, which is impossible with human. This is expected to provide precious information not only on antigen immunogenicity, but also on species specificity and on the kinetics of the antibody response. The next step was to check the suitability of R34K2 and AE34K2 in humans naturally exposed to Aedes albopictus in two areas of northeast of Italy, Padova and Belluno. The last validation of R34K2 antigen was in two areas with different epidemiological settings, where arboviral transmission is endemic, La Reunion Island and Bolivia. Thanks to collaboration with colleagues in Istituto Superiore di Sanità, 4 groups of mice were exposed to bites of Aedes albopictus, Aedes aegypti, or Anopheles coluzi. Blood samples were collected at different time points. 1 week before the first exposure, Baseline 1 week after the second exposure, midterm, and 1 week after the 4 last exposure, top, and then 1, 2, 3, and 5 months after the end of the exposure. The specific EGG response to SGA's ALE34K2, AE34K2 were analyzed by ELISA. Mice immunization was verified using EGG responses to salivary gland extracts of the corresponding mosquito species. As we can see from the graphs, all mice exposed to both Aedes albopictus and Aedes aegypti saw EGG response against a specific SGA. Also, we checked the possible cross-reactivity between SGAs. As we expected, there is a high cross-reactivity due to the common and relative conserved repertory of salivary proteins. First results with R34K2 were that 2 out of 4 Aedes albopictus exposed mice saw EGG responses to R34K2. In both mice, the response attained a peak 1 week after the last exposure and was stabilized 2 months after the end of the exposure regimen. Regarding to the Aedes aegypti exposed mice, all mice exhibited EGG responses to AE34K2, although at different degree and with slightly different kinetics. These results indicate that both AL34K2 and AE34K2 are immunogenic to mice. Interestingly, considering the relative high conservation of the 34K2 proteins in the 2 Aedes species, no immunocross-reaction was observed. This observation suggested that the 34K2 proteins from Aedes aegypti and Aedes albopictus may represent interesting species-specific markers to evaluate host exposure to these 2 Aedes species. The availability of a single human serum hyperimmune to Aedes albopictus saliva offered the opportunity to obtain some preliminary indications on the immunogenicity to humans of R34K2 and eventually on the immunocross-reactivity to AE34K2. An intense EGG response against both AL-EGA and AE-EGA was detectable at T1, confirming the hyperimmunization of the donor against Aedes albopictus saliva and displaying a wide EGG cross-reactivity to EGA from Aedes aegypti. On the contrary, the EGG response to R34K2 appeared considerably higher as compared to the response to AE34K2. At the time point T2, the EGG response to both AL-EGA and AE-EGA persisted, even though at slightly lower level. On the contrary, the specific EGG response to R34K2 had a remarkable decrease, and also levels of anti-AE34K2 EGG appeared reduced. Overall, despite the obvious intrinsic limitation due to the ability of a single human serum and by the hyperimmune status, this observation suggested that R34K2 is immunogenic to humans and that, as observed in mice, there may be a limited cross-reactivity in the 2 orthologous 34K2 proteins. We published this result last year in PLOS negative tropical diseases. So the next question was, are the 34K2 Aedes proteins immunogenic to humans in conditions of natural exposure to mosquito bites? For this purpose, samples from Padova as high-density area of Aedes albopictus and Belluno as low-density area of tiger mosquitoes were collected at different time periods, before and after summer, corresponding with the minimum and maximum density of Aedes albopictus. Collected samples and entomological data were available thanks to the collaboration with our colleagues from Istituto Zooprofilattico delle Venezie e Dipartimenti di Medicina Trasfusionale di Padova e Belluno. We first analyzed the EGG responses to Aedes albopictus EGA in sera collected before and after summer in the 2 study areas. Anti-EGA EGG responses were significantly higher in sera collected after the summer period, high mosquito density, both in Padua and Belluno, as compared to those collected before summer in the same areas. Moreover, EGG antibody levels against SGA were higher in Padua than in Belluno during both seasons. These observations confirm the original assumption of Padua being an area of higher exposure to Aedes albopictus than Belluno. After we analyzed the anti-R34K2 immunoresponse, when the different surveys were compared, a seasonal variation of EGG levels was found in Padua, and anti-R34K2 EGG responses were higher in Padua than in Belluno, both before the summer season. Comparison of anti-R34K2 IgG antibody levels between the two sets of sera collected in Belluno did not show a significant seasonal variation. However, when only paired samples from the two localities were analyzed, a significant seasonal increase was found not only in Padova but also in Belluno. Therefore, these observations convincingly suggest that IgG responses to R34K2 may be suitable to assess spatial and temporal variation of humans exposed to bites of the tiger mosquito Aedes albopictus. On the other hand, IgG responses to AE34K2 did not show difference between the two sets of sera collected in Padova and Belluno. These observations could be due to a low specificity of the AE34K2 salivary protein in areas where Aedes aegypti is not present. To get insight into the IgG subclasses specificity of antibody response to the R34K2 protein, we determined IgG1 and IgG4 antibody titers in the sera collected in Padua before and after the high-density mosquito season. As we can see from the graphs, when we analyzed both the whole population or only per samples, IgG1 titers are 10-fold higher than IgG4 levels. There is no time for a deep explication. However, we also analyzed the correlation between IgG response to R34K2 and AIDS, and it saw an age-dependent trend, probably due to the immunotolerance. And last, human cutaneous number of bites and time of bite were correlated also with IgG response to R34K2 in Padova and Belluno. We recently published these results in Frontiers, so the last question was, do individuals naturally exposed to Aedes albopictus and Aedes aegypti in areas when different epidemiological settings recognize the R34K2 and AE34K2 proteins? For this purpose, I went to Montpellier to analyze the specific IgG response to R34K2 in sera collected from adults exposed only to Aedes albopictus in La Reunion Island. To check the specificity of this biomarker for Aedes albopictus, cross-reactivity was tested by comparing IgG levels of anti-AE34K2 in individuals from Reunion Island. Also, IgG levels in Bolivian samples were measured to R34K2 and AE34K2 to check the suitability of this marker in individuals exposed to Aedes aegypti. The performance of this study was thanks to the collaboration with Dr. Frank Remou team in Ayardé. Individuals from France were the unexposed control group, and practically any of them responds to the R34K2. Contrary, individuals from La Reunion saw IgG response to R34K2, validating our antigen as a good marker in endemic area where Aedes albopictus is present. Also, there is a positive correlation between IgG response to AL-AGE, analyzed by our colleagues in Montpellier, and our results for anti-R34K2 response. On the contrary, we analyzed the IgG response to AE34K2 in the unexposed group from France, and we found a positive response. This result, in correlation with what we found in Padua and Belluno, could be explained by a non-specific response and may explain a cross-activity with salivary protein from other Aedes mosquito species. Bolivian samples respond positively to the AE34K2, even if the cutoff in this case is really high due to the positive response from the control group. The differences between both surveys are even more evident if we consider the seroprevalences. As we can see from the graph, the frequency of responders for the R34K2 in La Reunion Island is significantly higher than in Bolivia, 0.66 for La Reunion, 0.22 for Bolivia. Possibly, this seroprevalence in Bolivia for R34K2 may be explained by a low cross-activity with other Aedes species. Contrary, the frequency of responders for the AE34K2 did not show a significant difference between La Reunion Island and Bolivia. So, in conclusion, R34K2 and AE34K2 are immunogenic to mice and do not show cross-reactivity. The IgG response to R34K2 appears to be novel reliable candidate as a marker to evaluate spatial and temporal variations of human exposed to Aedes albopictus. We also validate R34K2 antigen in areas where arboviral transmission is endemic and maintained by Aedes albopictus. We check a low specificity and detectable cross-reactivity of AE34K2 in areas where Aedes aegypti is not present. And we also check a low cross-reactivity of R34K2 in areas where Aedes albopictus is not present. I would like to thank my tutor, Professor Brunner K, for the support and for leading me during this year, my team in Sapienza University, and all the people and institutions with which we collaborated. And thank you for your attention.
To Sara, brava, very nice presentation.
Thanks.
So, any question or comments?
I have one question.
Yes, please.
Hi. Ciao, Sara.
Ciao, Sere.
Do you know how different are the amino acidic residues of these two proteins from albopictus to Egypti?
I mean, the differences in the
sequence.
The positions.
Yeah, if they are different or conserved.
I mean, yeah, the sequence of the amino acid, the identity is a 62% between the homologous between Aedes aegypti and Aedes albopictus for the 34K2. But exactly the positions of the differences, I don't know. So I mean, I don't know if it's the conservative positions.
Okay, but I mean, they are quite different.
Yeah, exactly. For that reason. That reason, we go ahead with the homologous.
Okay, thank you, Sara.
You're welcome.
Okay, thank you, Serena, for your question. And I see a stand end, but okay. Just no. Anyone?
Posso fare una domanda?
Yes, of course. Please.
Oh, sorry, I was speaking English. Forget. Okay, congratulations, Sarah. It's very nice. No, my question is about the possibility that this kind of detection of exposure may have may provide some indication about climate change. I mean, in some cases, we know that the tiger mosquito spread through human activities, but the spread of Aedes aegypti can be sometimes connected with the climate change and global warming. And so I was wondering if this can be a field to be explored, to see if the coming out of exposure to Aedes aegypti can be in some way a early warning system for climate variation.
Yeah, of course, that is a good point, because nowadays we know that the climate change and is correlated, or the spread of this vector is correlated with climate change, because we know that we are no more in a temperate zone, it's almost subtropical zone, for example, Spain and Italy. So the mosquitoes are more adapt to survive in our temperature nowadays. So of course, yes, it could be another option to check through the antibody response to the salivary proteins of Aedes aegypti, places where it's supposed to be due to this climate change. Yeah, of course.
I think this could be also a good method to exploit research grants that are focused on climate change.
Okay, so thank you for your intervention and your question.